Unmute. Okay, so I say it again. I want to say good afternoon to everybody, and thanks for the opportunity. So, hue and chroma discrimination for natural objects. Color discrimination, but maybe more in general, color vision has been for long investigated with stimuli which are uniform. They are one point in color space. But actually, if you measure distribution of color distribution of natural objects, we did it with hyperspectral imaging for a large set of natural fruits and vegetables. When you plot the color distributions in color space, here is the AB plane of the CA-LNP space, what you see is that, of course, it's a distribution of pixels, and these distributions have specific properties. So if you focus on the cluster representing the orange, here each of those clusters is one of the fruits. You can see, it applies to all the others, that most of the variability is in what I call the chroma direction, so the radial direction towards the neutral pole. And then you have little variability in the orthogonal direction, which I will call hue. So there are a number of reasons by which this can happen. Already specular reflections can change chroma, also neutral reflections, but maybe the most prominent reason is shading, which belongs to most of the objects. And what happens with shading? If you have a simple Lambertian surface, like the example here with a sphere, shading scales up and down the reflected light of a factor that depends on the illumination angle, the angle between the direction of the illuminant and the surface normal, which is this term here. I hope you can see my cursor. So this reflects in this effect on the pixels in color space. So you have the best illuminated pixels, which are brighter, and then the more the illumination is parallel, the darker the pixels are, and also closer to the neutral point. So there is a correlation between luminance and chroma, which is due to shade. So this can explain at least in large, I would say in large part, the shape of the distributions we saw for natural objects. Of course, shading changes with shape and viewing condition. It's not really property here. It's not what makes us distinguish between objects based on the color. So hue is much more interesting. And in fact, the importance of hue might have been underestimated when you consider single point in color space. But if you look at distributions, like in the example I have here, the one I showed you before, even objects with very similar colors, like within the same quadrant in color space, when you plot, like here on the left, the hue histograms. So when we computed the LAB points and computed hue in our coordinates, and that's the histograms here, you see that it's easy to segregate objects based on the hue. Also, if you consider the color tuning of a D2 single cell, as indicated here by the circular data points and the fitted dash line, the tuning is around 0 degrees here and quite narrow as compared to what you would expect by the best fitted linear model that combines the conic responses, indicated by the continuous line. So there is, in physiology, a narrow tuning to hue, which would be useful to actually do what I did before, segregate objects depending on the hue, because the hue distributions are quite narrow. The importance of hue itself is not a new thing for perception. Judd already spoke about the super importance of hue differences. Subjective differences along the radial line in color space are smaller than those that would be expected from differences in the orthogonal directions, differences in what I call the hue. So for instance, if you have n steps along the radius, if the space is Euclidean, you would know how many steps you will have along the circumferences, 2 pi by n, but actually Judd argued it's twice as much. These dimensions are the same ones I referred to when I described the shape of the color distributions of each one, which is the one I show here on the right, color and hue. That's why we investigated discrimination for natural objects. We define our stimuli to vary in hue and chroma. One might say that hue and chroma by comparing couple pairs, but actually with this way we define the stimuli, we have comparable steps in the two directions, so we can actually compare pressures. We used 3D render stimuli because that way we could combine values and full control of the chromatic properties. For each stimulus, we specified the color as the reflectance defined in a point in color space. Like you can see, the reflectance corresponds to a point in color space, and you see that indicated here as the X, which happens to correspond to the brightest pixel, which is also the one with higher chroma. Then the rest of the pixels, because of shading, they move towards darker values and closer to the removal point. That's what we saw before with shading, so that we could create hue and chroma differences, so the stimuli different in chroma or hue, just by moving this reflectance parameter in color space. And we presented our observers with 4 stimuli. One was always the odd one, and they had to tell which one it was. And they had hue changes and chroma changes in 2 directions for hue and 2 directions for chroma in 2 quadrants, the first and the 4th quadrant, which I will call the purple and the orange quadrant. This is the kind of data we obtained. So class discrimination experiment with psychometric functions. We have a hue change in a certain direction, for instance, in the top left graph here on the x-axis. And then the bigger the change, the higher the proportion for the participant scale. We could fit this probability and infer thresholds for the 2 directions of hue change and for the 2 directions of chroma change. And I think you can already appreciate in this graph that thresholds for chroma will be bigger. And we could, of course, plot them in color space, average them, and that's what you see here. So these 4 points are averaged across 10 observers. And what you see for the 2 quadrants is that for the purple quadrant, thresholds are comparable. Whereas for the orange quadrant, you clearly have lower thresholds for hue than chroma. Is this the difference between hue and chroma can be already explained by the fact that you have more variability along the chroma direction, so by the shape of the color distributions of our stimuli. And this can be easily shown with a simple simulation in which I can take a very simple ideal observer that with the same stimuli presented to our participants, chroma and the hue changes, samples, let's say 100 pixels, compute the mean. Then you have 4 means, 3 from objects with different shapes but the same chromatic properties, and 1 after being changed PL for the hue and chroma change. And you have 4 means, and you can see, you can look at which one is more distant from the others, and that's what the model will tell. It's the odd object. With this, then, for each change you make in the KL space, you can compute the proportion of correct answers, you can fit psychometric functions and get thresholds. And the green curves are the results of this simulation. So what you can see is that hue thresholds are always smaller than chroma thresholds, but they do not differ between quadrants, and also the ratio between the hue and the chroma changes is closer to 1 for the simulation. So the ellipses in the human data are more elongated than simulated ones. Also, this works only if you have a distribution of points in color space, but it can't work for beautiful stimuli. So as a control, we took the same colors that we measure discriminations around, in this experiment. We presented our participants with uniform stimuli, like this one. And again, the same task. One is different, and they have to indicate which one. And you can see again the results of the discrimination experiment now in red with the dashed line. And thresholds are generally smaller, but the results are qualitatively the same. So you have lower thresholds for hue than for chroma in the orange quadrant, but comparable thresholds in the purple quadrant. Also for uniform stimuli. So the shape of the distribution is not the only issue. These results with uniform stimuli are actually similar to something that has been previously shown. Gauss-Krug and Gegenfurter measured discrimination thresholds around a number of points spanning the color circle. They measured in 8 directions, but now I focus on the ones we also measured to show that in the first quadrant, again, they found similar discrimination thresholds for hue and chroma, but hue thresholds were much smaller in the fourth quadrant. Overall, you can see that the hue thresholds tend to be smaller than chroma thresholds. Danilov and Mollo explicitly tested for differences in thresholds, in chroma thresholds and hue thresholds. So they measured in the 4 quadrants in the same axis that we did, or similar axis than what we did, but at different chroma. So they measured thresholds around points at different distances from the neutral point. Overall, what they found is, which is what they claim, is that hue thresholds are smaller than chroma thresholds. And this you can see in this diagram on the very right, where they plotted the ratio between chroma thresholds and hue thresholds. And you can see that for all the points they measured thresholds around, these ratios are higher than 1. Also, one thing you can see is that the further they measure from the neutral point, the higher the ratio. So given this property, we wondered whether our results, the asymmetry between the quadrants, was due to the fact that we were measuring around points with the same chroma, but maybe different perceived saturation, because it seems to matter the radial point at which we measure in color space. In fact, when Schiller, Valsecchi, and Gegenfurter measured isosaturation curves, like the ones indicated by the dotted lines here, you can see that in the KL space, these curves are not circular. So it's not that same chroma predicts same saturation color. For instance, if you take one point in the orange quadrant and compare to a point of similar chroma in the first quadrant, you would see that you would need twice as much chroma in the fourth quadrant for this point in the fourth quadrant to be perceived with the same saturation as the point in the first quadrant. So clearly there is pronounced difference in saturation. Question is whether this can explain the asymmetry between quadrants we found. So we looked at it empirically. We just measured thresholds around points and different chromas, spanning basically the whole chroma PKL. And these are the results in the same format as before. So you see the discrimination ellipses, in this case for uniform stimuli. And what you can see is that the eccentricity of ellipses changes with the quadrant, but not with chroma. So you see that again the same thing. That the ratio between the hue and the chroma differences is closer to 1 in the purple quadrant. So you find more elongated ellipses in the orange quadrant and more circular ellipses in the purple quadrant. Although when you go further and you measure around points which have higher chroma, you can see some elongation already also in the purple quadrant. So this data can be plotted the same as the histogram from all of it. And that's what I show here. And indeed, if you compare with data that is quite similar, you find that for the orange quadrant, the ratios between chroma and hue thresholds are always higher than for the purple quadrant. So always this asymmetry, no matter the chroma level at which you measure. And that's also what I found. And also you find this trend of increased ratio with the distance from one. So we found lower thresholds for hue than for chroma, and that's to notice that actually was never the case that chroma thresholds were higher. So that's a robust, across studies, strong effect. But these differences are more pronounced for the orange than for the purple quadrant. And the results apply both to natural distributions and to uniform stimuli. The superiority of hue on chroma discrimination and the asymmetry between quadrants does not depend on the distance from the natural point, because it's always there. I mean, there is some effect of it, but the asymmetry is always there. So we can look at it with different interpretations. It has been shown that the orientations of the discrimination ellipses aligns with the chromatic variation within the stimuli. And this is basically the same idea I introduced before, I told you before in the simulation. So you can assume that observers use some simple hue, like the mean color of the regions they consider part of the object, or the color of shaded regions. It's actually what a deep learning model proposed by Morimoto does to solve a color discrimination task for those objects. And but this, of course, works only if you have a distribution. So it won't explain results for the uniform stimuli, and also it doesn't seem to explain the difference between patterns. Another possibility is this correlated neural noise explanation. So if you can look at the diagram here on the right, it's in the Danilo Lammolo paper, this explanation. I forgot the reference, I'm sorry. So if you have uncorrelated noise on the two color axes, then the probability density of the signals that are elicited by color stimulus on these two axes is circular. And then no matter if you have a hue change like here or a chroma change, thresholds are similar, just depending on the noise. But if you have correlated noise, the probability distributions are elliptical, so that you can clearly grasp from this illustration that when you have a chroma change, there is more overlap, and therefore thresholds are high. But this doesn't explain the difference between quadrants. It works, of course, with uniform stimuli, because the noise is not out there in the world, but it's a sensory noise. But it doesn't explain the difference between quadrants. So here I just want to propose some idea. I won't show you any more data. What we propose is that, or maybe yes, maybe just one. We propose that one possibility, which is not an alternative, it's a complementary, is that both the difference between hue and chroma thresholds and also the symmetry between the quadrants are due to the fact that our visual system is tuned to the statistics property of the error. So if we look at our database of hyperspectral images of fruits and vegetables, and also at the reflectances provided by the Cambridge database of measured spectra, you can compute the hues, even daylight illumination, I think is 65. Then you here in this plot, I show you the distributions of hues. So where the portions of color space which are lighter are the ones where hues are more frequent. So you can immediately see from here that most of the reflectances we measure in the database are in the orange one. So one can argue that it's there that you find the difference between hue and chroma, because we want to tell apart, we evolve to tell apart objects that exist. But also, I mean, this tells why the orange quadrant is special, but it doesn't say why you find this distance also here in the second quadrant. It can be a byproduct of evolving the difference in the fourth quadrant, but maybe there is also some other factor, and indeed, Another thing that varies is not just the reflectance of objects, but it's the illumination. So, for instance, if you take one reflectance, like I have here, it's a message, and you see how this changes in the KL space by being illuminated by all the measured spectra from the Granada daylight spectrum database. So you multiply, you compare to BKL, and that's what you see in this diagram. The points are all along a line, which happens to be parallel to the daylight axis, which I'll show you here. That's a linear approximation of the daylight axis in BKL. So this would happen for each of the points in all the four quadrants. And if I repeat the simulation I told you before, but now the variability is not due to the shading or to having an object, but it's all the variability that you have by the illumination of the natural illumination changing over the day. And then again, you have this distribution and observe the samples, pixels. What you see is that you get the lower thresholds for hue than for chroma in the fourth and in the second quadrants, in the orange that we measured thresholds before and also here. But here you will get the opposite, you will get larger chroma thresholds. And if you combine this with what I told before, the fact that we want to tell objects apart, that won't make sense to evolve this shape. So this can explain why these ones are circular and these ones have this elongation. The interaction between these two needs, and objects apart and this kind of variability to them. Of course, these one are speculations, but they can be tested. Unfortunately, we haven't tested them again, but I tell you what we want to do in the last 2 minutes and a half. So we want to use a deep learning. We want to do a deep learning experiment or simulation. A simple approach would be to take one of these big and powerful networks, which are pre-trained on a large set of natural images to recognize objects. These perform in object recognition similar to humans, and that's why they're interesting as model of our perception. So the input data for these networks usually is these images, and the output is one vector that tells you the probabilities that it's one class of object, a dog or airplane. And so for our experiment, what we could do is to look at the representation in each of the hidden layers. So this representation consists of the responses of the filters that have been learned in all the layers. And we can look at all of them and basically simulate with a linear classifier the discrimination experiment that we had in human participants. So the input would be each of the four shapes. We have the same chromatic properties, just different shape, and one has been moved in color space. It can be a chroma or a hue change. And then in each of these hidden layers, you have a set of features. And it can be even per space. Some, depending on the architecture, it's possible that spatial structure is kept. So you have a number of dimensions that can be the input to a linear classifier. Then the classifier will be trained to tell which of the responses are elicited by the model. With this, there can be performance of the classifier, and therefore we can simulate the thresholds. We can measure thresholds for the network and compare it to the profiles with the humans. Of course, this will be done with uncalibrated images, and also there will be no control. It's just trained on natural or pseudo natural images. Now I know I have like 15 seconds or so. I will match, because what I want to say is that with computer graphics, we can actually have full control on the scenes we use for training, because we can photorealistically generate such scenes with illuminations and the reflectances we get. So we can have artificial illuminance, and then we can illuminate with both the natural illuminant or artificial illuminant. We can illuminate objects with natural reflectances, or with non-natural reflectances that cover the color space, like Manza chips. And with this, we could cross all these factors and understand the specific circumstances at which the discrimination profiles resemble what we found with humans. So we can have a better account for what was going on and solve the long-lasting problem. So to conclude, thresholds for hue are always lower than for chroma. These differences are more pronounced for the orange than for the purple quadrant. Results apply to both natural distributions and uniform stimuli, as shown before. And the superiority of hue on chroma discrimination and the difference between quadrants does not depend on the distances from the neutral point. There is a few interpretations that don't necessarily go against each other. And we want to develop this last one, that the discrimination profiles we observed emerge, at least in part, because our color vision is tuned to the statistical properties of the data. So we will look at it with the ends, and thank you.